Hello, my name is Samantha Baez, and I will guide you through my lipid pathway chart. Today we'll be focusing on lipid metabolism in pigs, which are non-ruminant animals. I've illustrated some relevant parts of their anatomy for reference, particularly the liver, which plays a crucial role in lipid metabolism. To begin with, let's cover the basics of lipid structure. Lipids generally consist of a glycerol backbone with fatty acid tails, which can either be saturated or unsaturated. In this case, the lipid has a phosphate group, which classifies it as a phospholipid. Lipids are known for having both hydrophilic heads and hydrophobic tails. Now let's move on to lipid digestion. The process starts with enzymes like lingual and gastric lipase, which break down triacylglycerols by hydrolyzing their ester bonds. These enzymes act at the SN3 position, converting triacylglycerol into diacylglycerol and a free fatty acid. Lingual lipase is secreted by serous cells in the mouth, while gastric lipase is released by the gastric chief cells in the stomach. As lipids enter the small intestine, pancreatic lipase becomes essential. This enzyme hydrolyzes ester bonds at the SN1 and SN3 positions, forming monoacylglycerol and two free fatty acids. However, pancreatic lipase requires bile for efficient fat emulsification and colipase to anchor it to the lipid droplets. Bile is a complex secretion produced in the liver and stored in the gallbladder. It contains bile acids, phospholipids, cholesterol, waste products, and inorganic salts. The bile salts, which are bile acids conjugated with taurine or glycine, reduce large lipid droplets to smaller emulsion particles. Pancreatic colipase acts as a coenzyme for pancreatic lipase, countering bile salt inhibition. Additionally, other enzymes contribute to lipid digestion, such as phospholipase A2 hydrolyzes phospholipids at the SN2 position to yield lysophospholipid and a free fatty acid, while cholesterol esterase breaks down cholesterol esters to free cholesterol and a fatty acid. In summary, lipid digestion occurs in three steps. First, lipids are emulsified by bile, forming emulsion particles. Second, the emulsion particles are hydrolyzed by enzymes, breaking down triacylglycerol into two monoacylglycerol and two fatty acids, phospholipids into lysophospholipid and fatty acids, and cholesterol esters into cholesterol and fatty acids. Finally, these digested products combine with bile salts to form mixed micelles. After digestion, the lipids are absorbed through the small intestine enterocytes in three phases. First, in the apical membrane transport, mixed micelles passively diffuse into the cell. Long-chain fatty acids are transported to the smooth endoplasmic reticulum with the help of fatty acid binding proteins through active transport. In step 2, within the smooth endoplasmic reticulum, these derived lipids are resynthesized into triacylglycerol, phospholipids, and esterified cholesterol, which assemble into chylomicrons. Finally, in the basolateral membrane transport, chylomicrons are packaged into Golgi vesicles, then released into the lymphatic system through excitosis. And unlike long-chain fatty acids, short and medium-chain fatty acids bypass the smooth endoplasmic reticulum and enter circulation directly. Bile salts, after their role in emulsification, are reabsorbed in the ileum and recycled back to the liver. And here's a diagram summary of lipid absorption. Next is lipid metabolism. Lipid transport via lipoproteins is very important, which deliver lipids to various tissues in the body. There are four main types of lipoproteins: chylomicrons, very low-density lipoproteins, and high-density lipoproteins. Each class transports specific types of lipids. For instance, chylomicrons carry dietary lipids. VLDL transports triglycerides from the liver, LDL carries cholesterol from the liver, and HDL retrieves cholesterol from dead cells. And here's an illustration of those lipoproteins and their components, and here is an illustration of lipoprotein synthesis, and here is the major pathway of lipid metabolism I'm about to discuss. Within lipid metabolism, triglyceride synthesis primarily occurs in the liver and adipose tissue, and this is closely tied to the liver's function in lipogenesis, where glucose is converted to fatty acids. In the process, acetyl-CoA, mainly derived from glucose, exits the mitochondria as citrate, and once in the cytosol, is cleaved into oxaloacetate and acetyl-CoA by citrate lyase, and through a 7-step process requiring NADPH, acetyl-CoA is transformed into palmitoyl-CoA, and stored glycerides are broken down through lipolysis, especially when blood glucose is low, insulin is low, and glucagon is high. In adipose tissue, triacylglycerol is converted to glycerol and free fatty acids. These free fatty acids enter the cytosol of energy-demanding cells, going through activation, transport, and beta oxidation. In beta oxidation, a 7-cycle process converts fatty acids like palmitate into acetyl-CoA, FADH2, and NADH, producing a significant amount of ATP through subsequent citric acid processing. In muscle cells, low insulin levels prevent the GLUT4 transporter from functioning, halting glucose uptake and shuttling acetyl-CoA into ketone body formation. Instead, these ketone bodies serve as an alternative energy source, especially during fasting. In summary, the result of lipid metabolism produces 129 ATP from just one palmitic acid molecule. Lipid metabolism is primarily regulated by hormones, specifically insulin and glucagon. Insulin promotes lipogenesis by activating lipoprotein lipase and GLUT4 transporter, while glucagon triggers lipolysis by activating hormone-sensitive lipase and inhibiting acetyl-CoA carboxylase. Additionally, citrate activates acetyl-CoA carboxylase, while malenyl-CoA inhibits CAT1, regulating the beta oxidation process. And here is a diagram to summarize what I had just discussed. Lastly, ketogenesis is a 4-step process that creates ketone bodies, which are acetoacetate, beta-hydroxybutyrate, and acetone from acetyl-CoA. In times of fasting, the heart and muscle tissues utilize acetoacetate to spare glucose for the brain. Your brain loves glucose, therefore it cannot use ketone bodies effectively and depends on that glucose for optimal function. Thank you.